Moving on, the next order of business, the next person I'd like to introduce is Dr. Marianne Wu in the National Institutes of Health. Marianne has been the program director or program manager, I forget the title at NIH, but for MAC-CHESS. We won't say how long, Marianne, because both of us couldn't be that old. Well, but the last, as many of you know, both CHESS and MAC-CHESS have gone through a lot of transitions in the last several years. And it's been Marianne's steadfast presence down at NIH has been a great benefit to CHESS and MAC-CHESS both. And so I'd like to thank her for all the hard work she's done. And I'm wishing her long health and long life and many more years together. So, so Marianne.
Thank you. And I'm sorry I was unable to attend during the day today. And one of the program officer duties is to listen to review meetings, and that's what I was doing. Just a few things I wanted to bring to update about NIH. We, as the world is responding to the COVID-19 pandemic in a few ways, and all of the information I'll be telling you, you can also find on the NIH homepage. There are two links, one for health-related information and another for grant-related information. And NIH has had a response for COVID research. Most of that is coming through the National Institute of Allergy and Infectious Diseases. At NIGMS, which is the home for MAC-CHESS's funding, we have two different mechanisms for grant competitive revisions. One of them is the SBIR STTR program with small businesses, where there is funding for therapeutic and diagnostic developments in the in that program, but you have to have an SBI or STTR to participate in that. The other area is in modeling of infectious diseases. We have a program called MIDAS, and any modeling for predictive modeling of infectious diseases, there's opportunities there. There's also a variety of administrative flexibilities for grantees. For instance, trainees and staff can be paid while at in-home, stay-at-home status. Grantees can donate PPE and other supplies. Late applications are accepted, and there are several notices that describe those details. If you're an ESI or a K99 fellowship fellow, you can have that fellowship extended. You just have to talk to your program officer and find out how to go about doing that. A lot of waivers are being given for prior approval and reporting extensions. So if that's an issue for grantees, we want to make sure that we make things as easy as possible in these difficult times. NIH and NIGMS are also putting together strategic plans for the next 5 years. And if you would like to look at the framework for those strategic plans or provide information that might contribute, like if you have concerns or interests in what directions those either NIH as a whole or NIGMS should be taking, there are requests for information where you can provide your input, and that you can either Google NIGMS strategic plan, or I can provide the notice. It's NOTGM 20034. And the other thing I wanted to bring up to date was resources in structural biology You may be familiar that NIH is putting a large investment in cryo-EM. There are 3 national centers that are located in New York, Oregon, and California. And they, just like McChess, was just getting up to speed. They just got their microscopes and they were getting ready to gear everything up when COVID hit. But eventually they'll have, each one of them will have 4 of these high-resolution cryos microscopes, and where NIH is encouraging the community to use these centers for data collection and training in structural biology using single particle analysis. And now we are going to be funding a new area of cryo-electron tomography, where tomography of cells and subcellular components will be funded through a network whereby 4 centers are being proposed to be funded for developing the samples for cryo-ET, which are a little bit tricky. You have to do things like focused ion beam milling and plunge freezing and knowing exactly where you're looking within the cells so you can get the high resolution data. And then 1 center where the data will be collected. So those are going to be funded in the next, within this fiscal year, which is by September. So that's something that you might want to know about. And as always, we look forward to good things coming out of McTest, and we're excited that you'll be reopening. Or maybe you have. I'm not sure where we are in that. I know it's June. Here we are in June.
Tomorrow morning.
Excellent. All right. So I don't know if you have any questions about that or anything else here.
Are there any questions for Marianne?
Okay, thanks.
Thank you, Marianne. So now the keynote speaker for the evening is Rick Cerione, and he's the director of MacChess. And I can tell us the project he's working on is both sprung out of his cancer research originally. He realized then it would apply to COVID work. It also makes unique use of some of the new techniques that MacChess has been developing in namely room temperature serial crystallography. Without stealing all of his thunder, introduce Rick Cerione.
Okay, thanks. Let me see if I can successfully share my screen, which I think I can. Let's see, we go to, and let me hit this. Okay, so I should thank Elke and Joel for this very kind invitation to give the after dinner talk at this year's users meeting. The original title was going to be along the lines of drug discovery, drug development with the help of Chess and McChess. Elke pointed out that wasn't appropriately sexy enough, so that we had to have something like SARS-CoV-2 in the title. So the story will be about how we actually ended up studying SARS-CoV-2 or COVID-19. So by way of an overview, my laboratory, as Joel suggested, over the last, I'd say, 5 years, have been working towards developing new types of anti-cancer compounds, anti-cancer drugs, where we've been targeting metabolic enzymes that are absolutely essential for cancer cells to survive, to metastasize, but aren't very important in normal cellular context. And so in trying to do this, of course, what would be very helpful would be to have structural images of how these drugs bind to the enzyme targets. And this is where we've run into some challenges, because when we've done standard cryo-MX on 2 different drugs that differ by orders of magnitude in their potency, their ability to bind to the targets or inhibit the target activity, in these standard MX structures that we get, they look virtually identical in terms of how they bind. So this is where we're placing our bets on serial room temperature crystallography, working with Aaron and Arena and Mariam, because the hope is at room temperature, we're going to be able to tweeze out the differences, which, as I'll show you, can be pretty subtle, but make all the difference in the world as to whether a particular drug candidate is very potent or rather weak. And then in parallel, we've also now are embarking on these small angle scattering or SAX experiments, because we know that the target we're studying actually, when it becomes maximally activated, forms these long polymers or filaments. The reason for that is that it serves as a scaffold to bring the next enzymes in a metabolic pathway into a large complex. And so we would like to see, using SAX, how these polymers are forming. In fact, we first got evidence of this through SAX with Richard and Magda and Soren some years ago. And we're especially interested in seeing how different drug candidates affect that polymer formation. And then importantly, and this gets to the gist of the story, these same enzymes that we've been targeting in cancer turn out to be essential for virus infection and for SARS-CoV-2 infection. So as background and maybe a touch of historical perspective, so my laboratory, pretty much when I first came to Cornell, and since then, and that goes back some years, we've been interested in the signaling pathways in cells that cause cells to grow normally, and then to see those normal cells perform functions, including important roles in development, and then what goes wrong in those signaling pathways when disease or disorders ensue, and in particular cancer. So as an outcome of studying these signaling proteins, protein complexes, we started to think about using small molecule chemistry to try and correct the problems that these altered signaling pathways are causing in cancer. And the way we went about that was to set up a model system in our laboratory. So we were growing cells that are often grown in biological laboratories, fibroblasts, and we were introducing into these fibroblasts a very potent cancer-causing gene, or an oncogene, that was originally identified in B-cell lymphoma. And we had actually found some years ago that this gene encodes a protein that starts excessive signaling pathways that cause these normal cells to go down the road towards cancer. Now, there are a number of differences, obviously, between normal cells and cancer cells. One of the differences in cell culture is that normal cells are really pretty behaved and pretty respective of their neighbors. So as they start to grow and touch their neighbors, they stop growing. This is what's called contact inhibition. But cancer cells show no such courtesy, and so they pile up on top of their neighbors, and they form these foci of cells, which you can stain for and pretty easily visualize under the microscope. So here in the left panels, what I'm showing in the upper left, these are cells looking under the microscope, normal fibroblasts. You don't really notice much of anything. They're just nothing distinguishing. They're just growing there politely. But in the lower left panel, those are the same cells where we express this cancer-causing oncogene. You can see all these dark spots. These are foci of cells. So it was this transition from normal to what's called oncogenically transformed that we screened a library of small molecules to look for small molecules that would inhibit this transition, or at least make these cells on the lower left look like these in the upper left. And what emerged from that screen was a group of compounds, and one in particular that we ended up codenaming 968, which is shown here in the upper right. And as shown in this middle panel, that small molecule could pretty effectively make these cells that had this oncogene expressed look normal. And right from the beginning, it showed some attractive features. One was that it showed chemical selectivity, because a number of very structurally or chemically similar compounds were ineffective in being able to block that transition. And so what that suggests at first glance is that this compound has a very specific target. But what was even more attractive at the time was that under the conditions where this small molecule could cause these cells on the lower left to become these cells on the upper left, if you simply added that compound to normal cells, it had no effect on their growth, their survival, their appearance. And obviously, this is something you always hope for. If you're looking for a lead in an anti-cancer drug, you'd like to have something that's very specific for the cancer cells and not affecting normal cells, because as all of you are probably well aware, that's been a problem that's plagued chemotherapy from the very beginning, that many of the drugs and compounds being used are not only killing cancer cells, they're killing normal cells under different conditions, and this gives rise to all these types of side effects. And so, given this, then the question might be, if you considered me a cancer biologist, which was sort of an evolution, what is this cancer biologist doing being the PI of McChess for the last 15 years? And I know some of my colleagues and some of the directors have asked that question frequently. Now, the short answer is that I was strong-armed by Saul Gruner 15 years ago to do this, because the argument that Saul made was, well, NIH believes, and I don't know that he bought the argument himself, but NIH believes that this marvelous facility should be used to ask really big questions in biology. So they think they need a biologist that should be the PI. So that was Saul's argument. The truth was, in that time frame, my laboratory was starting to appreciate that these proteins and protein complexes that we're studying, if we were really going to understand their function, we had to get structural information. And especially as we started to think about trying to develop drug candidates, because we would want to know how those drugs bound to these different targets. And so I had students and postdocs that I was shipping around to various collaborators throughout the country. I sent one of my students to Germany to a very renowned X-ray crystallographer for a couple of years, so that they would come back and start really bringing the appropriate technology and get me up to speed so we could start solving crystal structures of these different proteins that we thought could be important in cancer. And ultimately, one of the structures that students in the lab solved, and also Qing-Chu Wang at McChess solved, turned out to be a target for this small molecule 968. And it's a metabolic enzyme, which I'll be describing in a moment. But this, this is the ribbon diagram depiction of a tetramer, because this enzyme, when it becomes activated, becomes a tetramer. This is the ribbon diagram depiction based on the X-ray crystal structures that were solved. So the individual subunits making up this tetramer are just shown in different colored ribbon. And what we learned were a few things. One, that when this enzyme is present in normal cells, where it's typically present in very, very low amounts and it's essentially inactive, it exists as a dimer. When it ends up being expressed in cancer cells at high levels, it becomes very activated and it's a tetramer. And so the reason that the tetramer becomes active is that when this dimer at the top in purple and green engages this dimer at the bottom in blue and yellow through these helical interfaces, there's a loop that is initially close to where those helices are that ends up moving. We call this the activation loop. And in each of these subunits, when that loop moves, it sends a conformational signal, if you will, to the catalytic sites in each of these subunits, which normally have a lid over it. When they receive the signal, the lid pops open, substrates come in, and now you can end up having catalysis. So what is this target? Well, it turns out to be a member of a family of enzymes collectively called glutaminase. And it turns out that in humans, there are two genes encoding glutaminase. So there are two proteins. One is called GLS, or sometimes called GAC, and the other is called GLS2. And again, while in normal cells, these are expressed at very low levels and are essentially inactive under almost all conditions. In cancer cells, these two different proteins are expressed very highly and very highly active. And in different cancers, you might get different of the two different members expressed. So for example, in triple negative breast cancer, it's almost exclusively GLS that's highly expressed. Whereas in estrogen receptor and progesterone receptor positive breast cancers, which make up incidentally about 70% of all breast cancer, it's almost exclusively GLS2. In some cancers, you get both expressed, such as pancreatic cancers, brain cancers. In colon cancer, it's pretty much GLS. In ovarian cancer, it's almost exclusively GLS2. So what are these enzymes doing? Well, they catalyze the first step in the metabolism of the amino acid glutamine. What they do is they catalyze the conversion of the amino acid glutamine to glutamate. And in a moment, I'll show you why that's important in cancer progression. But again, what's also important is that viruses infecting cells, SARS-CoV-2 infecting cells, cause these enzymes to be highly expressed, and they become absolutely dependent on the functioning of these enzymes. Okay, so in cancer, why are these enzymes so important? And the reason is that nearly 100 years ago, a German biochemist named Otto Warburg first found that cancer cells undergo a pretty striking change in their metabolic programs. And in fact, in the cancer biology world, this is referred to as the Warburg effect. And simplistically, all this really is describing is that in normal cells, which is shown here on the left, the metabolic program starts when sugar, specifically glucose, enters the cell, and it starts a sequence of enzymatic reactions in the soluble part, in the cytoplasm. This is called the glycolytic pathway. And the end product of that sequence of enzymatic reactions is a metabolite, pyruvate. That metabolite then goes into the energy organelle of cells called the mitochondria, and it starts a cycle of enzyme reactions called the TCA cycle. And each enzyme in that cycle, each reaction is generating molecules of ATP, which are the basic energy molecule of all life. So all normal cells satisfy their metabolic requirements, their bioenergetic needs through this coupling of the glycolytic pathway in the cytosol to the TCA cycle in the mitochondria. But things change in virtually all cancer cells. That is, the glycolytic pathway is uncoupled from that cycle in the mitochondria. That, in fact, is what Warburg first saw. But that ends up posing a bit of a dilemma for cancer cells because they need a feed into this cycle. They need to turn that cycle, not so much for making lots of ATP molecules, but to produce building blocks for biosynthetic processes that cancers need to be cancer cells. They need to be able to survive different stresses that normal cells would never be able to survive, including breaking out of an initial tumor site, getting into the circulation, surviving, and getting to a secondary site. So they need something to compensate for the uncoupling and the feed that normally would have come from this pathway, glycolysis. And the way they compensate is by greatly increasing their ability to metabolize this amino acid glutamine. In fact, are often said to be glutamine addicted. And that occurs as an outcome of a greatly accelerated first step in glutamine metabolism, this conversion of glutamine to glutamate by these enzymes, GLS or GLS2, that are very highly expressed, highly active in the cancers. And again, the target for the small molecule 968. And then the product of that reaction, glutamate, converted by another enzyme into another metabolite that now feeds and turns the TCA cycle. So this is how glutamine addiction enables cancer cells to satisfy the metabolic requirements that they need to end up being cancer cells. So then a question would be, so how does this small molecule 968 bind to these enzymes and block their activation? So what I'm showing here is based on or space-filling depictions of two subunits of the enzyme in cream and gray. And then the other two subunits, if it was forming a tetramer, are shown in purple and green. So this top red circle is circling a cove in that dimer that forms. And the small molecule 968, which is depicted here in green, nestles into that cove. And when it binds in that cove, it subtly changes the conformation of this dimer so that it can no longer receive signals that are coming in cancer cells, and specifically in cancer cells, that cause some post-translational modifications that enable this dimer to then become an activated tetramer. So the reason that this small molecule is so specific for cancer cells and not normal cells is because they're blocking signals that this enzyme would only be receiving in cancer cells. And just as a frame of reference, this bottom circle is circling a red molecule. This is a substrate analog that's in the active site of this gray colored monomer. And so you can see the 968's acting from afar. It's acting allosterically. And let me give you just another view of this if I can. So here I'm showing a dimer of the enzyme in red and yellow, and I'll try to use my pointer to give you a view of that cove where the small molecule fits in. Should be coming into your view in your plane in a moment. I'll try to get the pointer right in there. It's right in that hole. And so you can see it was really by serendipity that we were looking for small molecules that would inhibit signals that were being caused in cells by a very aggressive oncogene. And what we end up finding are a group of compounds, and one in particular that can manage to find its way into a cove of an enzyme that's absolutely essential for the survival and ultimate metastatic spread of those cancer cells. So we've been developing analogs of this small molecule 968, and what that required is that we develop a direct readout for the binding of 968 or analogs to the pure recombinant enzyme. And the way we did that was to place a fluorescent reporter close to that cove so that when 968 or an analog binds to the pure enzyme, it causes a quenching in the fluorescence of the reporter. So we have a readout as a function of dose, and we can develop binding isotherms. And we also developed a spectroscopic readout for enzyme activation. So now, in the same cuvette, we can simultaneously look at 968 or analogs directly binding to this protein and then how that affects the enzyme activity. So an example of that's just shown in this lower left panel here, where the y-axis on the left, we're just looking at the changes in the fluorescence as 968 is binding to this pure enzyme. The y-axis on the right is looking at the percentage inhibition, and all this is a function of 968 concentration. And you can see there's this beautiful correlation in a dose-dependent way between the ability of this compound to directly bind to that protein and the effects that it has on enzyme activity. Now, we've developed a number of analogs, some of those shown here, and what we're plotting in the lower right, the y-axis is the equilibrium dissociation constant, which is a measure of the affinity of these different compounds for that protein target. On the x-axis, the IC50 is just the half maximal concentration that you get inhibition. So it's a measure of the inhibition of enzyme activity. And again, you see this really nice linear correlation. But the other thing you'll notice is that it's spread over some range of doses, which means, again, some of these molecules are more potent than others. And this gets to the crux of the question. We really want to understand what dictates potency to continue to make better compounds. And it's pretty subtle. And I'll just show you an example of that. So here, these are some of those compounds, and I've simply classified them into either good inhibitors, which means they're relatively potent, or bad inhibitors, which means they're pretty poor. And we're just going to look at these top two. So in the top left, that's 968. And one of the things we had found is that this ring and this bromine is especially important in the ability of that compound to inhibit the enzyme. Now in the top right, this is a compound that we originally named WPK968 because it was synthesized in the lab by a postdoc, William P. Catt. Of course, the journal reviewers made us change it to a number, so now it's 335. But it's virtually identical to 968. It has that bromine. In fact, the only difference is this nitrogen here in this ring, which we initially thought shouldn't have much of any effect. Turns out that changes the potency by 25, 30-fold. And so it's really very subtle, and it keeps getting to this question. We want to understand what is it and how they bind so that we can continue to make more and more potent drugs. Now, it turns out that this enzyme we've been targeting, there's a second class of allosteric inhibitors. So here on the right, I'm just showing again in space filling the tetrameric form of this enzyme. And so here, the top half in light and dark blue, that's one dimer. There's the cove, that's 968 trying to nestle in there. The bottom half, light and dark blue, the other dimer coming to make the tetramer. There would be another cove down here. The lead compound for this second class of inhibitors is called BPTES. This was actually first identified by a biochemist at Colorado State for totally different reasons. And then a biotech company on the West Coast, Calithera, started making analogs of this compound, changing mainly the ends of the molecule. And they had one they named CB839, which they reported to be much more potent than this lead, BPTES. And in fact, this is now in clinical trials for triple-negative breast cancer and multiple myeloma. And then Bill Catt designed analogs that mainly changing the middle roughly of the molecule. And he has analogs, one of the UPGL0004, which is also much more potent than BPTES. And in fact, this is more long-lived in animals than CB839, which we think suggests it would even be a better drug. Now, these compounds bind in a distinct way from this 968 class of inhibitors. They bind in the interface where these two dimers come together to form a tetramer. You get two such compounds bound per tetramer. And when they bind there, they kind of lock in that activation loop I was talking about, preventing it from moving. And if the activation loop doesn't move, you don't get the signal to the active site for the lid to open, so you don't get catalysis. So these compounds essentially trap the enzyme in an inactive tetramer. Now, like the case for this 968 class of inhibitors, we've continued to be developing analogs for this class as well. So we've developed a direct binding assay, putting a reporter group again close to where they bind. And we've also can read out, of course, their effects on enzyme activity. So here I'm just comparing two compounds. This is the lead, BPTES, in the closed circles. This is in open circles, the compound Bill developed. And what we're looking at is the inhibition of enzyme activity as a function of drug concentration. This is a semi-log plot. And the point here is that the dose curve in going from the lead to Bill's compound is significantly shifted to the left. This is a semi-log plot. So this is a 50-fold difference in potency. And this really gets to the crux of our challenge, because Qing-Chu Wang has solved the crystal structures for both of these compounds bound to the enzyme GLS, and they look identical. So again, the reason why we have to get other methods to be able to tweeze out what makes one drug much better than the next. Now, if we take the 968 class of inhibitors, or if we take this class of inhibitors and introduce them into mouse models for different cancers, we can pretty significantly inhibit tumor progression in those models. But what we've been doing more recently is thinking about developing cocktails of combinations of metabolic inhibitors with others. The reason for this is that one of the real challenges in cancer therapy has been that a given drug is being administered to a patient, after some months, the patient's cancers or tumors stop responding to the drug. And the reason, or at least one of the reasons, is that the drugs that have been developed mainly by the pharmaceutical industry that are targeting different steps in the signaling pathways that are telling these cells to grow with abandon, to be cancerous, those are being directed at different points in such a pathway. But cancer cells are very devious. So with time, they can develop other pathways so they can get around that block and achieve what they want to achieve. And so what we have been thinking about in other groups as well is if we can combine some of our metabolic inhibitors with compounds that would stress the tumor cells at the initial tumor site. If you stress them, then what happens is those cells become extremely dependent on metabolic reprogramming, extremely glutamine addicted, because they need those building blocks to be made to withstand the stresses. And so I'll show you an example of this. So our hope is that if we can stress those cancer cells sufficiently and then come in with the metabolic inhibitor so you can't get the building blocks, we can whack the cancers early and maybe get around the problem of resistance. So this is just a set of experiments where we were testing this sort of idea. So what we were combining in these experiments is the metabolic inhibitor 968 with Avastin. Now Avastin was developed by Genentech some years ago as an anti-angiogenesis drug. That is, it blocks the ability of blood vessels to form at the sites of the tumor, and the blood vessels are forming there so they can deliver nutrients to these tumors, and the tumor cells need that to be able to grow as rapidly as they do. And in fact, Avastin, it's FDA approved, it's been through clinical trials, it's been in cancer treatments, although a few years ago it was pretty much rescinded from breast cancer treatment because as a single treatment agent, it really wasn't, for different limitations, showing a significant benefit. So what we hope is that if we combine Avastin with these metabolic inhibitors, we can achieve a benefit. So here's a set of experiments, and we've been doing this in what are called patient-derived tumor graft models for breast cancer. So basically you get tumors from breast cancer patients, you can graft them, you introduce them into the mice, you wait until these tumors are starting to appear, and then you can either allow them to grow in the animals untreated or you treat with compounds of interest. So these are triple negative tumor grafts. And what we're looking at in the left panel is just on the y-axis, the size and growth of the tumors as a function of weeks in the animals. So in black, those are animals where the animals are untreated. And after about 2 weeks, those tumors are really starting to take off. In blue are animals where we started to treat with Avastin, and it's giving some slowing of the growth of those tumors compared to the animals that were not treated, but it's not a dramatic slowing. Now in green, these are animals where we started treating with a very low dose of the metabolic inhibitor, 968. And in fact, when it starts, this actually causes the tumors to take off a little bit. But eventually they relax and they end up growing pretty much at the rate of animals that weren't treated at all. So there's certainly no benefit. Now, as we increase the dose of 968 in these animals, we can start blocking tumor growth, but not at this level or this dose. However, if we combine this dose with this level of Avastin, we get this red line. We really get a significant synergistic inhibition of the tumor growth. We've done this in a number of these graphs. This is just another set of experiments where in this case we cut out the tumors after about 8 weeks. So in the upper left, those are the control animals or the tumors from those. And then in these panels, they're just single treated animals, but you can see the combination really has a significant effect. And we can achieve this if we combine the other class of metabolic inhibitors as well with Avastin. And that's all we're showing here. So again, these are triple negative tumors. And in this case, we're combining Bill's compound UP4 with Bevacizumab, which is just a formal name for Avastin. And again, you see in black or green and blue, they're all pretty much not doing much to affect the tumors. But if you combine those two compounds, you really get a dramatic inhibition. So it was that result that got this investment group, Kairos, to start investing in drug development to see if we could really do something in developing anti-cancer drugs. Now, there's no question that we have a long way to go here. So going from mice to humans is a big jump. So there's still a number of experiments, but at least now we're getting some financial influx to help us with this development. Now, this then brings us to this question. So where does SARS-CoV-2 fit into all of this? And to do that, we have to at least say something about, so how does SARS-CoV-2 infect cells? And I realize many of you may already know this. And in fact, this I simply lifted, I've shown this before, from Time magazine, because this was there so to help, as they state, with the lay public understanding how this virus infects. So in this depiction in red is the virus SARS-CoV-2. There's an envelope that's made up of lipid molecules, and these are basically the structural features of all biological membranes. And then within the envelope are proteins, and then one of the proteins is jutting out at regular intervals, and it looks like a spike. So this is called the spike protein, and in fact, it's because you get this spike-like appearance that gave the name to this family, coronavirus. And then within the virus are RNA molecules. So when this virus approaches a cell that it might infect, and this could be a cell in your nose, it could be a cell in your lungs, it can be in your intestines, it binds to a protein that's on the surface of those cells. So protein is performing a normal, important physiology. It's called the angiotensin converting enzyme 2, or ACE2. This enzyme is a transmembrane protein that's present in a number of different cells, and it's there really to help maintain normal, proper blood pressure. But it turns out ACE2 is able to bind to the spike proteins on SARS-CoV-2. But that binding only occurs after the spike protein has become activated. So these cells that are getting infected are not only providing a receptor, ACE2, that's there for other purposes to bind to the virus, but they're also secreting enzymes, specifically a protease, that has other roles, but is able to clip a piece of the spike protein away. And now what happens, that enables the spike proteins to bind more tightly to this receptor, ACE2, on the cell surface, and it enables this envelope of the virus to fuse with the cell membrane so that essentially it can now inject the proteins, the viral proteins, and the viral RNA into the cells. Now, once that happens, those proteins start changing the metabolism of the cells. They make that cell now glutamine addicted, and the proteins together, the metabolic changes, together with the RNA, which is being replicated, starts to make additional viral particles. And as these viral particles start to accumulate in this host cell, eventually, in most cases, that cell is going to die, it's going to break open, it's going to lyse, and it's going to shed all these viral particles to go and infect other cells. So the simple strategy here is that, as I told you earlier, normal cells, their metabolic program is dictated by the coupling between the glycolytic pathway and the cytosol in this TCA cycle in the mitochondria. In cancer cells, that coupling is disrupted. So cancer cells cause the high upregulated expression of these enzymes, GLS or GLS2, and they become glutamine addicted. And these are the enzymes, again, that we're targeting with our so-called drug candidates. Well, the same thing happens in these virus-infected cells. This pathway gets uncoupled, these enzymes get highly expressed, these cells become, once virus-infected, glutamine addicted. So the argument would be the same anti-cancer drug candidates we're developing should work against these virus-infected cells. Now, there's one other similarity between cancer cells and virus-infected cells. And it has to do with this famous drug, hydroxychloroquine, which has been in and out of the news for quite some time. This is an anti-malarial drug. It has some antiviral capabilities. And at one point, it was hailed as the miracle drug for COVID-19. And as you all know, one of our leaders was taking it prophylactically. Now, it turns out that when it was being used extensively in hospitals treating patients who are in pretty bad shape with COVID-19 at doses that are typically used with malarial patients, they were seeing some pretty frightening side effects, particularly with COVID patients who had a predisposition to cardiac disorders, as this was giving rise to arrhythmias. But we had found well before this pandemic that hydroxychloroquine can block the building blocks that are formed in response to the metabolic reprogramming of cancer cells. And so that had got us thinking about, would there be a benefit against cancer in combining hydroxychloroquine with our metabolic inhibitors? And what we would find, in fact, is that we could get a really striking synergy. And I'll just give you a quick example of that. So here what we're looking at is a really nasty brain cancer cell that we're growing. And this brain cancer cell really doesn't respond to almost anything we've tried, certainly as a single treatment. So this blue histogram is just showing that they're growing and surviving perfectly well when not treated. In red, we were treating with just 968 at this level, not really touching it. In green, we were treating with hydroxychloroquine at this level, not really touching it, but look at the two together. We really get this dramatic synergistic inhibition of these nasty brain cancer cells. And so what it suggests to us is that if you use the right combination, you might be able to sufficiently drop the dose of hydroxychloroquine so that you don't have to deal with some of the side effects, but you can still get maximal benefits. And so it was this data that got us fast-tracked at Cornell to start doing SARS-CoV-2 or COVID-19 research. So where are we now and where are we going and where's McChess and Chess and all of this? So we have members of the lab in molecular medicine who are developing what are called pseudoviruses. So these viruses, for all intents and purposes, look like SARS-CoV-2. They have the right envelope, they have the spike protein, they're able to bind to ACE2 on the right host cells, they'll infect the cells, they change the metabolism. So we're using that system, and I should say, but they're missing, these pseudoviruses, a component that prevents them from heavily replicating so they're not infectious. So you can do these studies in a BSL-2 facility, a standard cell culture facility. So what we've been doing is looking at those different metabolic inhibitors that we have, trying to see how they alone in combination or in combination with hydroxychloroquine can affect the ability of these pseudoviruses to infect the host cells and change their metabolism, and comparing all that to host cells that haven't been infected, because what we're zeroing in on are the best conditions where these drugs or drug combinations will specifically inhibit the glutamine-addicted virus-infected cells. And then we're giving these conditions now to a virologist who's at the vet school, Gary Whitaker, who's one of 20 sites in the US who's growing the actual human SARS-CoV-2 in his BSL-3 facility. And so he's taking what we think right now are our best conditions to see how they affect a real human virus. Now, I should say a couple things. One, these are really tough experiments because it's very, very difficult to maintain cultures with these viruses in a very reliable way so you can really do drug analysis. And this, in fact, was one of the great problems with HIV. But we have an extraordinary graduate student who finally has been able to achieve this. And Gary's now also achieved it. So we're hoping soon we're going to get at least our first round of results And we think it's going to be an iterative process, of course. So he's going to see something, it's going to come back to us, we're going to manipulate, it's going to go back to him. So where does Chess and McChess come in? Well, we have postdocs in chemistry working with Qing-Chu Huang, and they're growing crystals of these enzyme targets with different drugs bound to them. And we're hoping with Aaron and Arena and Marion, we can use serial room temperature to really learn something about why a given drug is much more potent than another. And then in parallel, we're doing the Sachs experiments with Richard, because as I mentioned earlier, this enzyme, after it forms a tetramer, it will, to become maximally activated, continue to make long filamentous polymers. And so we want to look in Sachs to watch how they form, and we want to especially see how different drug candidates affect that polymer formation, because we think this could give us really new insights into what drugs are really better than others. So in a very complementary way, we may learn from Sachs which are really the best drugs we want to look at, and then from serial room temperature, why those best drugs are the best drugs. So I think we have really exciting opportunities in front of us, and that'll go well beyond these 3 weeks or so of experiments we're starting to embark upon at Chess. I've always said that independent of whatever happens with our drug candidates and whether they work against a virus, we're hoping that these methods will really represent new ways to do rational drug design, which, as many of you probably know, has had its limitations over the years. And that if we can really achieve that, then other investigators can come to the synchrotron with their targets and their drug candidates. I've also been pretty excited by the fact that for the first time in 15 years, as PI of McChess, I have a significant part of my laboratory that's truly embedded in the new developments that are happening at this facility. So with that, I'm happy to end. Let me just point out a couple of people who have been really important in what I described. Ray Lee, she's the graduate student that single-handedly has gotten our lab up in doing virology with some help from this postdoc, Kai Green. And then Sean Milano and Ram Ramachandran are the postdocs in chemistry who are working with Qing Chu to grow crystals of these target drug complexes. And then, of course, I'm indebted to Aaron, Richard, Arena, Marion, Chris Conley, Joel, Elke, everyone at Chess and McChess, the entire staff, indebted to Marianne Wu for letting us still have money. And with that, I'm happy to end.
Thanks, Rick. You have a question from David Schuler. He said co-crystals versus soaks.
Co-crystals versus soaks. So I think what they're doing in the lab now is mainly doing soaks. But I mean, Qing Chu might answer that. I mean, over the last year, we've tried both, but I think what they're mainly doing now is soaking in the drugs.
And Hector, you can go ahead and talk.
Dude, remind me never to come after you in a conference. That was fantastic.
Yeah, right. Yeah, right, Hector. I'm really worried about that, Tito.
And and don't tell Trump about the hydroxychloroquine that at low dosage can actually be good.
Don't worry.
No, this was absolutely awesome, my friend.
Yeah, well,
if I were younger, I would ask if you had.
Long way to go. These are tough experiments. So
I realize that, but if I were 30 years younger, I would ask if you had any space for an upcoming graduate student. Actually,
come on, we'll let you do a sabbatic. Come on.
There we go.
Yeah.
Fantastic, dude.
Thanks, Hector. Ken, you can go ahead. You might have to unmute yourself. There you go, Ken.
Okay, great. This is Lois, who's with Ken right now. Hey, Rick, beautiful talk. I'm wondering if you've used MD simulations to help understand how the different drugs bind to the protein.
Yeah, Ram is really interested in that, and he's done some. There's nothing that he's done so far that I would say has changed our thinking, or at least given us an insight. Now, part of it may be he's self-taught in doing those simulations. So, but that's certainly something he's been very interested in and he's been trying to do. So.
Okay, thank you.
Thank you. It looks like that's it. Oh, we have one more. Saul, you can go ahead.
Rick, sorry to be a nudge,
Yeah,
but
since you strong-armed me into this 15 years ago.
is there any difference in the RMS deviation of the alpha carbon backbone with and without the drug bound?
Yeah, I know. You asked me that question, I think. And there's subtle differences in the region of where I talked about that activation loop.
Yep.
There are some subtle differences there. And so one of the questions is, so the one class of inhibitors binds very close to that activation loop and actually we think sterically prevents it from moving, which prevents the conformational signal. But the question is, what about this 968 class that binds some distance from that region?
Mm-hmm.
Nonetheless, their binding affects changes in that region. So there are differences, but we don't understand how that's being transmitted. I mean, we presume there's some sort of structural change. And it gets complicated because we think where that 968 class of inhibitors bind further up on the molecule are the N and C termini, which have to kind of cross over, and we think they send a signal to that activation loop. So we think we're perturbing that by the binding of 968. But the bottom line answer is, yeah, there are some changes there.
Are those changes smaller if you look at the inactive drugs?
I'll tell you, those change, yeah, but I wouldn't, but what we don't know is why in the chemistry does that happen? So why a given, like the WPK 968, why does that compound, which seems to be less potent, seems to bind less effective, and admittedly does seem to give less changes there? Why is that? I mean, so all of this, I mean, what we'd like to be able to know is what is it about these chemicals or the chemistry so we can make better drugs and get potencies that are more like 100 nanomolar rather than close to 1 micromolar.
Great talk, Rick. Thanks.
No, thanks. Yeah. So I guess that's it, huh? Thank you all.
There's one more question, Rick. Sorry, Nozomi, you can go ahead.
It's not a question, Rick. I just want to say that was awesome, and Tito did not pay me to say this.
Yeah, well, thanks. Yeah, no, but one of my students did. We won't get into that.
Okay, I just like a couple of closing remarks.
I mean, she didn't ask. Oh, that was the question.
You got off easy.
Thank you, Nozomi.
Thanks, Rick, for a great talk.
Yeah.
And I guess in closing tonight, one, I'd like to congratulate all the poster prize winners and the paper prize winners, and fantastic job. Thank Rick for the talk. I'd also like to give my heartfelt thanks. This is the first time we've had a remote user meeting, and there's an army of people, well, not including me. I did not do this. An army of people made this possible, organizing the user meeting, all the remote access and so on. And thanks to all of them. They've been working incredibly hard for weeks. You see some of their faces coming by on the screen. But thank you, everybody, because the one thing everybody knows is I didn't do anything. And so thank you very much to all the people who are making us look good. So with that, I'd like to thank everybody. The sun is just going down outside my window. It's been a beautiful day, and I hope to see you all tomorrow morning. Good night, everybody.